So our next speaker is Chen Meng from TUM, Germany. He will talk. He will present activity landscapes of cancer cell lines predict drug response.
Good afternoon, everyone. I would like to invite you to explore the Atlantic together with me. It's a short for activity landscape of tumor cell lines. Can I use this? Good. So in this project, we measured the proteome and phosphoproteome of more than 100 tumor cell lines. There are two panels. One is 65 colorectal tumor cell lines. Another one is NCS 60. Consists of 9 cell lines from 9 different tissues of origin. And we identified in total more than 10,000 proteins and more than 50,000 phosphorylation sites. The goal of this project is to identify potential protein and the phosphomarkers that can predict drug sensitivity. Therefore, we included more than 1,000 drugs from public domain. So the first analysis we did, we call it hypothesis-driven model. There are some drugs. We know the proteins that are related to the mode of action of these drugs. As an example, we selected 5-FU. And this is a drug widely used in the treatment of colorectal cancer. It's a prodrug. The active form relies on a series of metabolic processes. There are a few tens of proteins involved in this process. We select those proteins. And using CRC65 data set as training set, applied random forest to train models. And apply these models to predict drug sensitivity of NCS 60 cell lines. As expected, the model trained on the CRC65 performed well in the colon cell lines in NCS 60. But something surprising here is that the models also perform well for the leukemia cell lines. So there is a potential of repositioning 5-FU to the treatment of leukemia. In the follow-up experiment, we selected the leukemia and colon cell lines that predicted to be positive, resistant, and sensitive, and did in vitro experiment and validated our hypothesis. Of course, when you have such a large data set, you always want to find biomarkers in an unbiased way. I think for this purpose, the elastic net already becomes a standard method to do, and we also used it. It recapitulates known proteins. For example, Schlafen-11 is known in the mRNA world. It is a sensitivity marker for many DNA reagents. Here we confirmed it on the protein level. As another example, the peripherin, an AKT1 inhibitor, and we find a few phosphorylation sites from proteins that interact with AKT. There are many examples like this, and sometimes we know, sometimes we don't know, but it makes a lot of sense. So one of the problems in the analysis of phosphoproteomic data is the missing value problem, and there are much more missing values in phosphoproteome than mRNA data or even the full proteome data. So we use two different methods to solve the problem. One is to sum up the phosphosites from the same proteins to the protein level. So we call it phosphoprotein abundance. And of course, there are some sites don't have a good correlation with the phosphoprotein abundance. We keep them separate. So at the end, we have two matrices. One is the phosphoprotein abundance. One is the raw intensity of sites. And also, we think instead of matching, associating individual sites to individual drugs, we can try to associate a group of sites to a group of drugs. So we can average out them to some biological, average out to the truth of biology. But we can use PLS to do this, but here we have two matrices. We used an extension of PLS. We found concordance analysis is which accept more than one matrices as independent variables. The result is promising. Actually, we found that many of the components are driven by the drugs that have the same mode of action. Here is an example. We find 3 drugs have highest score are the ESR inhibitors here. ESR is very important in the treatment decision in breast cancer. You can see here. The markers we selected are here. Actually, this is a loading matrix. And we found PGR, progesterone receptors, as sensitive marker, and also GREB1, which is known related to the function of ESR. Next, we want to know which cell lines actually driving the component. We found 2 breast cancer cell lines, MCF7 and T47D cell lines, very sensitive to the 3 ESR inhibitors. And of course, here we are using the phosphocyte in the model, and we want to know, could we find the same information on the protein level? You can see here the protein level of GREB1 and PGR are expressed in many other tissues in the 60 cell lines, but for the site information, they are more or less exclusively expressed in the two breast cancer cell lines that are sensitive to the drug. In the clinical practice, standing PGR protein is well established. In our cohort here, the PGR high expression is a marker that predicts long survival time, and we want to know if we include the phosphocyte, does it add anything on top of it? So we selected those PGR positive patients and further standing the phosphocytes. And actually, it also stratified patients into 2 groups, and the group have low expression of the phosphorylation site of PGR, have a shorter survival. So actually, it does add some value. So conclusion, we have a comprehensive collection of proteome and phosphoproteome of large number of cell lines. We can use them to find potential biomarkers, predict drug sensitivity. And also, the hypothesis-driven model helps repositioning the drugs. Benefit from the depth of our data set. 5FU example is just one of the examples, and it could be applied to many other drugs. And also, we created new computational method to overcome the missing value problem, particularly in the phosphorylation side, phosphoproteomics. So last, I would like to thank all people in our chair, and also our collaborators, the core facility, Bayern MS, Clinical University Hospital Frankfurt, and the Klinikum ESA. And the last thing, we are always actively recruiting bioinformaticians and computational biologists in our lab. If you are interested, you can contact me or write emails to our professor, kusta@tum.de. Thanks.
Thank you.